All set.
All right. Hi, welcome everyone to another Biostat Department Lightning Talks event. I'm Harris Reeder. I'm first year in the PhD program, and yeah, welcome. So our first speaker is Bob Glynn, who will be giving us an amazing talk. So, take it away.
That's hopeful. Much of my job over the 28 years I've worked at the Brigham and Women's Hospital has been in monitoring large clinical trials, investigator-initiated clinical trials. I want to talk about the particular challenges related to the last one. I spent 6.5 years writing data monitoring reports for. It's a trial called CANTOS, funded by Novartis, testing and what made the trial complicated from a monitoring perspective was three things. It was multiple doses, it was pleiotropic effects, and it was in individuals who had, we had almost no prior experience in human populations with this drug, and what we had was not related to the indication we were studying. So it was a trial testing the anti-inflammatory hypothesis in cardiovascular disease. It gave a human monoclonal antibody, IL-1 beta antibody, that was only approved for rare indications, kids with rare inflammatory conditions. It's given injectably every 3 months. And the trial randomized individuals into 4 groups, a placebo group on the right, or 3 different doses. There was no 3 different fixed doses of the target drug in individuals who already had heart disease. 10,061 people randomized in 39 countries. It ran for about 6 years. It was an event-driven trial that needed to accrue 1,490 primary endpoints, which was what's called MACE, major adverse cardiovascular event, and with a key secondary endpoint that also added unstable angina requiring unplanned revascularization. So my job was to monitor, writing reports quarterly for an independent data and safety monitoring board. It was it's pre-scheduled efficacy and futility analysis, but safety monitoring regularly because concerns existed and were noted, and that made how you report and put together the information in the absence of strong priors, no experience with this drug and these kind of people. Off-target inflammation pathway is quite diverse, involving adverse events and all kinds of body parts, and it made what happened? Here we go. Here's the timeline of the trial. Randomized subjects first in 2011, followed till 2000, and the trial finished last year. Primary endpoint results were reported in Barcelona at the European Society of Cardiology. A surprising off-target cancer benefit was reported separately in the Lancet. Mediation analyses have also been reported. At the time of the first futility analysis, there was serious consideration given to stopping one of the arms. The drug didn't look terribly effective for a major component of the endpoint, cardiovascular death. You can see there was a 19% non-significant higher rate of cardiovascular death in the active versus placebo group. And there was a highly significant trend for more sepsis infection deaths. And the curious, lacking a clear mechanism, at least in the eyes of the DSMB, that was unaware of a considerable literature on IL-1 beta blockade and cancer incidence, and particularly cancer metastasis. So one might have had the prior that the cancer death was real, but the committee mostly thought that it was chance. and they were really serious concerns. They let it go, and we need ways to display the many potential areas where adverse events might occur, and we took to plots showing the pooled active groups versus the placebo group. Here are the most frequent serious adverse events sorted by the log of the hazard ratio. You see about half of them towards harm, about half towards benefit, and the two that were most strongly towards benefit were an expected reduction in osteoarthritis, as it is an anti-inflammatory drug, and a very surprising reduction in lung cancer deaths. It was quite specific to lung cancer, actually. We also showed trends and wondered to what extent the trend evidence, that's my death sign. So we also looked at trends and as ways to display what was going on to a DSMB. We looked at AEs as well as SAEs in similar patterns, and saw often as many beneficial as negative. Some of them grouped on the harm side in expected ways, biological effects on neutropenia, leukopenia, not unexpected. And the primary result was this trial was reported as a positive trial. It did meet its endpoint for the intermediate dose only, met the multiplicity adjusted threshold for significance, 15% reduction in the primary, and there was a complicated gatekeeping approach that passed some alpha onto the key secondary only for the 150. It was positive. There was this very surprising but really strong dose response reduction in cancer mortality, driven mostly by lung cancer mortality, and that's all I got to say.
Any questions for Dr. Glynn?
So this was not dose escalation, right?
No, I misspoke. It's fixed dose. You were assigned to one of 3 fixed doses of active therapy or to placebo, and you kept that dose during the 6 years of your treatment.
Any speculation as to why the highest dose was only effective?
It was that, I wasn't so clear. That was not the highest, it was the penultimate. It was 50, 150, and 300.
Right.
It was the 150, the intermediate dose. They actually gave more alpha to the intermediate dose because they thought the high dose would have more side effects, and it didn't. It didn't. It did have more sepsis issues, but it also had a greater benefit on lung cancer. So the 150 and the 300 had about the same hazard ratios, about the same 15% reduction. More alpha was allocated to the 150. It met the threshold. The 300 did not. But they were the same effect.
All right. Thank you very much. All right. I forgot to mention that Scott Carter is unable to talk today, but next up we have Pete Kraft.
So it really is the good, the bad, and the ugly.
Sorry, what was it?
I don't know, fire...
Where's Valerie? Where's Valerie? Oh, is she outside? She got them on here. Let's see.
It's just called Kraft Lightning, I think.
Maybe she's
It's called Kraft Lightning.
Oh, it's on the desktop.
Where is it?
There.
Where is it? Oh, thank you. Sorry.
That's pretty good.
Like emotionally. Does that play that? Oh. I have no idea. I think it's like Was it short? Do I have to do anything? No, I think it just goes. Yeah, she's coming.
Okay. Great. All set?
Yeah.
All right. Thanks, everybody. I'm really excited to give a talk in this series. All the previous talks I've seen have been fabulous, and I'm hoping not to screw that up. So I'm Peter Kraft. I'm a professor of epidemiology and biostatistics. I'm the director of the program in genetic epidemiology and statistical genetics. And many of you may know me because I spend a lot of time analyzing GWAS data and gene-environment interaction data. But today, I want to talk about my lab's work in the broader space of integrative genomic epidemiology, where we take data on social context, behavioral exposures, biomarkers, lipid levels, metabolomics, and we try and put it all together in order to better understand disease biology and to build better prediction and prognostic models. So in terms of better understanding disease biology, there's a story that Nate Silver tells in his book comparing building models for predicting the weather versus models for predicting earthquakes. And the earthquake folks say they do such a crappy job because they can't measure the relevant stuff. Like the things you want to measure are miles and miles and miles underneath the surface of the planet, and you can't actually measure it directly. So it's really hard to build these models. And I think for observational epi, we're in a very similar position. So we can measure genotype and exposures, we can measure phenotype, but we don't really know what's going on sort of under the surface, under the skin, right? And there are 1,001 different causal mechanisms that we could sort of cook up that are consistent with that observed pattern that we see. So if we really want to understand which of those 1,001 stories is true, we're going to have to collect some more data. We're going to have to get under the skin, we're going to have to get better measurements of the environment, and we're going to have to put them together in order to tell a better story. Of course, once you start doing this in the real world, you run into all sorts of issues in terms of data quality, dimensionality, dealing with temporality, all kinds of weird confounding issues, collider bias. So these are things that we think a lot about, and some of the papers in this space are that folks in our lab have worked on are listed on that page. So the better understanding disease biology, I'm totally confident we're going to get there through these integrative genomics approaches, but it really is a long-term thing. Like this is basic science. It doesn't just pay off right away. I have a wish list of tattoos. This is the first one on the list. So I don't know. You'll see me sporting that soon. But in terms of things that we can do more immediately, we can actually use some of these results to build better prediction models. We don't have to understand what's going on under the surface as long as we have a good predictor. So this is some work that Richard Barfield, a former student here, did with Joel Cryer, who's at the Brigham and Women's, sort of showing the distribution of risk in the population as defined by genetic markers for, I think this is breast cancer. And it's we're sort of different lines over time. So back in 2007, this is prostate cancer. We knew of 3 markers, so there's some discrimination, but the line is pretty flat. But as we discovered more and more markers, the line gets steeper and steeper to the point where you can actually identify 5% of men who are at almost 3-fold increased risk. And that starts to become maybe clinically useful, especially when you combine it with other data. So thinking of breast cancer, if you think of family history, just looking at family history and measured germline genetics, so about 0.2% of women without a family history are at greater than 3-fold average risk. But if you focus on women with a family history, you measure the same genetic variables, you can identify 10% of women almost who are at greater than 5-fold average risk. And that's getting to be the point where you might actually intervene and do something to reduce the risk. Of course, just saying that you might be able to do it doesn't mean that it's a good idea. So one of the things that we're doing are some micro-simulation economics models to sort of figure out, compare different screening strategies. So here is the current recommendation from the US Preventive Services Task Force. Every woman starting at age 50 should get biannual mammograms. So you could think of another scheme where everybody at, say, age 30 gets their genetics measured, and depending on what your result is, you get put in one of these different arms. So for a very small percentage of women who are at greater than 5-fold relative risk, I'm illiterate. So if you're greater than 5-fold risk, then you would start getting screened at age 30 every year. If you're at less than a half average risk, then maybe you can wait to start at age 45 and you only have to come back every 3 years. So if you compare those two strategies, the number of life years gains relative to not screening at all for the current guidelines are 118 versus 196 for the stratified approach, at a minimal increase in terms of overdiagnosis and false positive tests. So I want to close really quickly because I feel like I have to. We're defending GWAS because you've probably heard in the lay press, in the scientific press, and even in this room that GWAS had been a failure. So just bottom line, this is what we knew about the genetics of breast and prostate cancer in 2006. We knew BRCA1 and BRCA2, of course, for breast cancer, and that was basically it. For prostate cancer, we knew nothing, despite trying very hard. And here's where we are now. We've basically 2/3 we've identified or have a pretty good feel for what's going on. So we haven't solved everything. Certainly we didn't do these studies and suddenly the scales fell off our eyes and we suddenly understood exactly what makes people get cancer. But we've got a lot of really interesting leads in terms of biological hypotheses, and we can do this rich stratification that I just talked about, which you couldn't do before. Thank you.
All right, any questions for? All right, great. Thank you. And next up we have Dr. Jamie Robbins.
There are only 3 people on the list.
Not three?
Yeah. Running the analysis now.